characteristics, their classification, and type study. That means we are going to discuss about Elphidium and protozoan diseases. So protozoans are the most primitive forms of animal. So they belong to animal kingdom, but they are very primitive. They appeared at the very early stage on Earth. So it was coined, the term protozoan was coined by Golfas. Okay. So proto means first, zoon means animal. So it's the first animal to appear on Earth. That is initial. So about 50,000 different types of protozoans are present, identified till now. So all over the world, we can actually identify protozoans. And for the existence, like if we see bacteria, fungi, how uh, their environmental conditions. Suppose like some requires like if they in the moisture condition, fungus will grow more. So like that, even protozoans to live, moisture is very much required. It is necessary. So majority of protozoans are solitary. That means they live alone, not in the form of colonies, but they live alone. Some are they live in colonies, and they are mostly parasites. So parasites are the animals which live on inside the host or out on the host, so that they can utilize the resources from the host and they can grow. And some of them also use humans as their host. So they are very important. So the important protozoans, especially they cause some diseases or they live in human beings are Leishmania donovani, which causes Kala-azar, okay, or visceral leishmaniasis, and Entamoeba histolytica. So it's very common one we used to heard like since our school days. So it is an amoebic dysentery. So it's again a human parasite. Then Plasmodium vivax, okay, which causes malaria, and trypanosoma, so which is a hemoflagellate, and they live in blood, lymph, and other tissues of the vertebrates. So they causes trypanosomiasis or sleeping sickness. So these are very important because they cause some or the other kind of diseases in the human beings. So they are the parasites which live in human beings. So what are the general characteristics? So they will have some common general characteristics. Like most of the protozoans are small in size, which are microscopic. You cannot see with your naked eye. They are colorless, and few are colored, but we cannot see most of them using naked eye. And most of the animals belongs to this phylum are free living as a colonies or sorry as a individual, and or they can be parasitic. So the body structure is very simple. They are unicellular. They have only single cell, and all the physiological functions are performed by that single cell itself. And the body symmetry is either bilateral. Bilateral means which is divided into two halves, okay, or spherical, round in shape. Some may don't have any symmetry at all, okay, rough, like irregular shape. And the body is naked. That means it is not surrounded by any plasma membrane in some protozoans. While other will have some kind of like pellicle, thick pellicle. Usually we see on insects, we see this pellicle. Okay. Some may have calcareous covering. That means made up of calcium or silicus. Okay. That is silica particles, some kind of shell. So this is for the protection. If we, especially in parasitic protozoans, for their protection from the host, usually they have this kind of arrangement. And body consists of protoplasm. They have one or more nuclei and ecto and endoplasm. Okay, and the function of locomotion, so they have to move, they have to feed. So all these is mainly by pseudopodia, that is false finger. Pseudo means false, podia means finger. So a finger like projection, or using flagella or cilia, and their nutrition is different in different animals. Okay, so like they may be holozoic, holophytic, saprophytic, parasitic, or mixotrophic. That means they can feed on the host, parasitic. Okay, uh, holophytic or holozoic. So saprophytic, they may live on dead um, organisms, or they take it from the organic matter. Okay, mixotrophic, so they can have mix of two or three type of nutrition. Then digestion is completely intracellular within the cell in the food vacuoles. And for respiration and excretion, there are no specific organelles present. So through the outer surface of the body only, this respiration and excretion takes place. And osmoregulation in freshwater forms is through contractile vacuoles, which serves as excretory organelles. Okay, mostly food vacuoles, excretory vacuoles, these are present. Then reproduction can be either asexual or sexual. Okay, asexual, it is mainly binary fission, simply dividing the parent cell into daughter cells, or multiple fission or budding. Is a proportion will come out of the mother cell. Okay, so that is budding. And sexual reproduction is mainly by conjugation. Okay, so a bridge is formed. So the genetic material passes, then fusion of the genetic material takes place. So you know conjugation, right? So through conjugation. So life history is marked by alternation of generation in most of the forms. Like that is life history will be changed like alternatively in the generation. Then encystment, so a cyst will be formed. Okay, under unfavorable conditions, whenever there is no food or any food material is not present. So in such cases, a cyst will be formed to protect itself. So it will remain dormant for the favorable conditions. And once the favorable conditions appears, so again it will burst the cyst and it comes out to be vegetative form. So now these protozoans can be further classified. Okay, and this classification was introduced by Hyman in 1940. So this recently, this protozoa was included in the kingdom Protista along with algae and fungi. Okay, but earlier it was in the older system. Okay, now the classification is based on the mode of locomotion, that is movement, and sexual and asexual reproduction. So this phylum Protozoa is divided into Plasmodroma and Xylophora. Two. Okay, these are subphylum. So this Plasmodroma is again divided into Sarcodina, Mastigophora, Opalinata, Sporozoma, and Celiophora is further divided into Ciliata. So this is class. So phylum, subphylum, and class. So all the protozoans in the subphylum Celiophora, they have cilia and they perform their movement based on the cilia. Whereas based in the plasmodroma, the cilia is absent. So that is basis of the locomotion. So based on the locomotion, they have divided into plasmodroma and celiophora. Okay. Now we will discuss each class. So first is masticophora. So as I said, some terms will be repeating. So I will be just skipping the terms. I don't explain. I will read and already whatever I have explained. If any new terms comes, then I will discuss. Okay. So masticophora, they are free living or parasite, and they are called flagellates. So flagella is present. Okay. They are free living in fresh water, sea water, and moist soil. And parasitic forms are found in the tissues of the host animal. So whatever the animal, it can be human being also. So in that and different tissues they will be occupied. And the body with a definite shape, it has a pellicle which is covering, external covering, made up of cellulose or chitin or silica. And they have flagella, one or more flagella. This is mainly for locomotion and food capture. So they are monomorphic nucleus, only single nucleus, which are single form of nucleus. Okay, morph means form, single form of nucleus, monomorphic, single nucleus will be present basically. The nutrition is holophytic, autotrophic, they synthesize. Holozoic, which is heterotrophic, they take it from the organic matter. Then saprophytic or mixotrophic from dead organisms. Then sexual reproduction is in some groups by syngamy. Okay, and about 2000 species are present in this class, and they are again subdivided into based on their mode of nutrition. They are phytomastidina and zoomastidina. So in the phytomastidina, so they are euglena. Example are euglena, whereas zoomastidina, it is mastigoamoeba. Mastigoamoeba. So this is about the first class. Then second is Sarcodina. Okay. So they usually live in freshwater, seawater, and moist soils. And majority live in free living, but some are parasitic. And the body is naked. It is not covered by any plasma membrane or pellicle or anything. And some will have shell around the body. Pellicle is absent. So they have pseudopodia, that is a false finger-like structure. So, which is used for locomotion and food capture. And this is present during, flagella may be present during some stages of life. And it is holozoic, that is heterotrophic in nature, the nutrition. So heterotrophic means from organic or inorganic matter, they usually take up the food, breaking down of the organic matter. Okay. Asexual reproduction is by binary fission or multiple fission or spore formation or budding. Okay. Then sexual reproduction is by syngamy. It's a fusion. And that is more than 8,000 species are identified. Again, they are subdivided into two classes, subclasses, which is Rhizopoda, Actinopoda. So examples of sarcodina are amoeba, entamoeba, then Arcella, Elphidium or Polystomella. So in here, in the case study, we are studying about Elphidium. Okay. The next is class Opalinata. So here they are endoparasites. Endo means inside. Parasites. They live in the body of the host. And they are found in rectum of cold-blooded vertebrates like frogs, toads. Okay. And they are completely and uniformly ciliated. You can see the image. So the cilia is present and which is completely covered all over the body and it is uniformly present. And they are saprophytic mode of nutrition, that they feed on dead matter and that the nutrients will be transported throughout the cell. It has two or more nuclei, and asexual reproduction is by binary fission and sexual reproduction is by forming gametes. So they form cyst in the breeding season of the host. So example is Opalina. Then sporozoa. Spora means seeds, zoa animals. So here spores, they form spores. Okay. Especially they have internal parasites in the vertebrates, arthropods, worms and mollusks. So they have thick pellicle around their body and locomotory organelles or cystosomes or vacuoles are completely absent and they are saprozoic or holozoic in nutrition. And asexual reproduction is by multiple fission. Sexual reproduction is by spore formation or syngamy. So where infective stages of life cycle are formed in sporozoites. Okay. Here in Plasmodium, especially malarial parasites, we can see here. So life cycle shows alternation of generation. So generation is like one generation is this form and the other generation is the other form, like that. So over 2000 species are known, which have divided into 5 subclasses and 10 orders. So examples are Monocystis, Plasmodium, and Sarcocystis. Then Ciliata, okay, also called as Infusoria. So it is the only class in subphylum Celiophora. So its presence is based on the presence of cilia. So cilia is present all over the body, and the body is covered by firm pellicle. So which cilia is used for their locomotion, that is movement and food capture. They are usually found in freshwater or seawater. Most of them are freshwater and some of them are parasites. So here they have mouth, cystosome, which we call, and gullet. This is cystopharynx, like what you call gut. Okay. Then holozoic mode of nutrition is seen. In some of the animals, these bundles of cilia, they form a filter-like structure through which the food particles are filtered. Okay, because it has cystosome, which is mouth, which acts as mouth, and cystopharynx. It creates a water current, which carries the food material. Okay. So two types of nuclei are present, macronucleus and micronucleus. So macronucleus is for the vegetative activity and micronucleus is for the sexual reproduction. So asexual reproduction is by transverse binary fission or budding, and sexual reproduction is by conjugation or autogamy. So here more than 5,000 species are present identified in this class, and here four subclasses. They are divided into four subclasses and 15 orders. Examples are Paramecium, Vorticella, and Balantidium. Okay. So here, so this is about the general characteristics of the different classes. Now the type study is Elphidium. So we will be discussing in detail about this Elphidium. So here it belongs to, or also it is Polystomella. Okay. So it belongs to phylum Protozoma, subphylum Plasmodroma, class Sarcodina, order Foraminifera, and genus Elphidium. Okay. So it is the shelled protozoa because it has a shell. Okay. And this shell are perforated. The perforation means it has pores. Okay. So it is usually present as a marine form. So in the marine water you can see, and especially in the depth of 550 to 600 meters under the sea. And brackish water also we can see this, and it is called as bottom dweller because we see that at the bottom of the sea. Okay. And it is a free living animalcule. Animalcule means small animal. Okay. It moves slowly along the sand, seaweeds, and debris of the seafloor. So it's a very minute organism. Okay, we can see, but it is very minute. So under the microscope, it is like a snail shell. Okay, and it has granular protozoa. So granule, small particle-like thing will be seen in the protoplasm. Protoplasm is nothing but like a cytoplasm. Okay, then Elphidium shows dimorphism. Oh, sorry, here I have missed something. Yeah. A distinct ectoplasm. Ectoplasm is completely absent, and a contractile vacuole is present here. And it has dimorphism. That means it can occur in two distinct forms, that is microspheric and megalospheric. So what is microspheric forms? Here, microscopic are less abundant. We don't see many number of animals in this form. They're smaller in size, micro means small, multinucleated, and the nucleus is small. And the initial chamber or proloculum is small, initial chamber, and it reproduces mostly by asexual reproduction, but that is by fission. Whereas megalosporic, they are abundant. Most of the animals are seen in this form. They are larger in nature. They have only single nucleus. Okay. The initial chamber is very big. Okay. And it reproduces sexually. So here you can see A and B. So microsporic and megalosporic. Yeah. So you can see the difference in that. So the shell is present, which is very hard, biconvex and oval or spherical shape. It is pale yellow in color, so it surrounds the protoplasm like cytoplasm. It is made up of calcium carbonate and other salts, and it is multilocular, many chambers, and each chamber is like V-shaped, anterior convex, and inside posterior concave. Okay. So adjacent chambers are separated by a septa, which is called wall, which has holes, pores, okay, which is perforated. Small pores are present, and this protoplasm flows from one chamber to another through these pores, okay. And the central part of the shell is called as umbo, and the peripheral parts are called keel, okay. And the shell surface has large number of perforations because perforations holes are mainly for movement of protoplasm, okay. And it has long pseudopodia, which is for movement. Then the cytoplasm or protoplasm, okay. So the shell chambers in Elphidium are filled with the cytoplasm, which is it flows from one chamber to other chamber through pores. So within the cell, the cytoplasm is distinguished into outer ectoplasm and inner endoplasm. And they have streaming movements like streams. They move like a water stream. That is called a cyclosis. This help in the distribution of the nutrients and also waste. It can carry the waste from all the chambers, and also it can distribute the nutrients throughout the cell. And it has mitochondria, ribosomes, Golgi complex, endoplasmic reticulum, all these organelles. And their size is based on like microspheric or megalospheric. And so microspheric has several nuclei, where megalospheric has only one nuclei. Okay. But no special organelles are present for the excretion. And excrete products are usually diffused from the surface of the body. Okay. Then pseudopodia, which is long, slender, and branching. So they are called as mixopodia or reticulopodia. They are for locomotion, that is movement and capturing food material. So what are the food habits and nutrition? So here usually they stick to mucus-like surfaces, and the food particles are carried from the water current and through the pseudopodia, and they have digestive enzymes present in this mucus layer, so they digest this food. So this process occurs entirely outside the shell. Okay. Then in the process of digestion, these food materials or nutrients, they enter into the endoplasm of the reticulopodia, and then it is distributed or circulated throughout the cell. Then respiration and excretion. So especially respiration is by simple diffusion of gases in and out. And the nitrogenous excretory material like ammonia or any other nitrogenous compounds, they are excreted outside the cell. Then reproduction and life cycle. So here it has asexual and sexual mode of reproduction. So asexual reproduction is by multiple fission. Okay. And here such microspheric individual is called as agamont or shigont. And sexual reproduction occurs by in megalospheric individual or gamete. So how this asexual and sexual reproduction takes place, we will look into it. So this asexual reproduction, so the microsporic form usually they undergo asexual reproduction through multiple fission or schizogamy. So here the cytoplasm flow out of the shell and forms a halo, that is completely empty shell, and the nucleus breaks down into chromatin bodies, and again they reorganized into nuclei. First nucleus divide, okay? They form nuclei. Now a bit of cytoplasm surrounds each nuclei, form amoeboid cells, which are called as amoebulae or agametes, okay? Then they detach from the parent cell and they create another shell, okay? And the first chamber of the shell becomes the initial chamber and it grows later. First it forms and then it grows. Then this protoplasm enters into the chamber and finally it settles down in the middle chamber of the shell. Now, completely they are formed. They have uninucleate, large, megalospheric adults once it is formed. Okay, in other words, microsporic form gives rise to megalospheric forms by asexual reproduction. So first, this microscopic forms, they first everything goes out the cytoplasm, then they divide nucleus first, then new shell is formed, then cytoplasm again enters. They first form the microsporic form, then they form megalospheric form through asexual reproduction. Now, once a megalospheric form is formed, it reproduces sexually. That is called gamete. So here the nucleus of the gamete divide mitotically. You all know mitosis process, right? So mitotically divides and into many nuclei, and the cytoplasm breakdowns into small fragments. So now each nuclei is surrounded by small proportion of the cytoplasm and a pair of flagella. So these bodies are called gametes or zoospores or flagellae. Okay. So now all the gametes are called isogametes or zoospores because all are similar in their shape and structure. Now they escape into water through pores and they will be freely swimming in the water using flagella, and then they slightly elongate. Now the haploid gametes of different parents, they fuse, okay? They fuse to form diploid zygote. This is called as isogamy, okay? Now each zygote secretes a shell around itself and becomes a young microscopic individual, and again new chambers are formed, nucleus divides, and now initially by meiosis, then the cell divides by mitosis called schizogamy, and now several haploid nuclei and the other microscopic individual, which is cyzoid, is formed. Now you have a microsporic individual. Again this microsporic individual, again it forms megalosporic individual, right? So through asexual reproduction. So this mode of reproduction is different in different animals. Okay, it can alter. Some may don't have any meiotic division, some may have mitotic division, like that. And the life cycle is defined as a regular succession of the various stages or phases by which the organism, it continues its propagation. Okay. So here this microsporic form reproduces asexually to form megalosporic form, and the megalosporic form reproduces sexually to give microsporic generation. That point you have to remember. So this is the image. So once you go through, first you write down like step by step and you can correlate with this image. So that will give a good idea. So all this entire life cycle, it completes about 2 years. Okay. Now we discuss about various protozoan diseases. So first one is Leishmania donovani. So this is named after the scientists Leishman and Donovan. So both have simultaneously reported the organism Leishman. So Leishman from London and Donovan. He's from Madras. So during 1903 only, both of them individually they have reported. So based on their name, that was given. So it causes Kala-azar and visceral leishmaniasis. It's a common disease in India and other eastern parts of Africa and South America. It was a public health concern in 1940s, and it is endemic in Assam, West Bengal, Bihar, and eastern states of districts of UP, Tripura, Orissa. Okay, so this parasite usually live in endothelial cells. Okay, in the spleen, liver. So endothelial cells they line the tissues, right? So in the bone marrow, and these parasites they generally reside in the leukocytes of the blood, spleen, liver. Leukocytes are white blood cells, right? Lymphatic glands, bone marrow, and they are also found in cats and dogs, and they can be normal or normally present, or it is acting as a reservoir host, okay, which will spread the disease. It is like an intermediate form. So it has two stages, leishmania form and another is leptomonad form. Okay. So leishmania form occurs in man and leptomonad form occurs in gut of sandfly. So here, so leishmania form, it has the colorless microscopic parasite is found or oval in shape, and the body is surrounded by pellicle, and the cytoplasm is not divided into ectoplasm or endoplasm, and a spherical nucleus is present. Flagella is okay, and these structures will give rise to flagellum such as blepharoplast or parabasal body or rhizoplast. Okay, the structure of the flagellum, it has these parts: blepharoplast, parabasal plast body, and rhizoplast. So, which is the root of the flagellum. So, in this stage, they are present in the endothelial cells of the vertebrates, and it divides by binary fission, and the cells are packed within this with these parasites. Around 50 to 200 or even more may be found in the affected cell. They attack fresh cells by multiplication, and then this is taken up by the sandfly. So here in the Leishmania donovani, it is digenetic, requiring two hosts for completion of its life cycle, and its primary host is man and secondary host is sandfly, which is blood sucking. Okay, and here Phlebotomus argentifus is the secondary or intermediate host which carries from one to other, and the reproduction occurs by asexual mode of reproduction, that is binary fission. So once from the human being, from the endothelial cells, it enters into the sandfly. So through blood, this sandfly serves as an intermediate host where the blood-sucking fly sucks blood from the infected patient, and now the parasite enters here of the alimentary canal of the sandfly. Where the leptomonad form or flagella takes place. Gut of the sandfly, it will form a spindle shape. It becomes large. It has flagella, which is small in size, and then it has a membrane. There is no membrane here. They have this parasitic form is short, free flagellum. Okay, small in size. This is the infective stage of the flagellum. So here, the sandfly is not affected by the presence of, it is just a carrier. Again, when this sandfly bites another person, another human being, now this parasite enters into that human being. So now the inoculation into the human being takes place where it will enter into the leptomonad form. Then it again changes into the Leishmania stage. Again it divides and it gives more number of parasites. So what happens when it multiplies? So the incubation period is about 3 to 6 months. That means we don't know till 3 to 6 months. So it will divide and all, it will spread. So it is also called Kala Azar, and this is systemic, that is only particular region, especially liver, spleen, bone marrow, and other viscera. It has irregular fevers, weight loss, reduction in number of white blood cells, enlargement of spleen and liver. The skin color turns black. Okay, it becomes fatal if not treated. Usually, if we identify in the initial stage, if it gets treated, it will be treatable. Okay, cutaneous leishmaniasis. That means cutaneous means skin related. So it is by another species of the leishmania. So here lesions like pimples kind of things are formed, then ulcers, ulceration on the skin, legs, feet, etc. And even that can heal spontaneously for many months. So it may not appear even after that. The repetition of the disease is very less common. Okay. So how to prevent this? So we have to like eradicate sandflies, spraying of insecticides in the house and other, if you know, like the areas where it is more spread, then adult flies, eliminating breeding place of sandflies, like cracks and the other parts of the building, then removing shrubs, vegetation of in the human dwelling, then poultry, all this. We have to take care if it is nearby. And proper medication, if identified early, proper medication should be taken to control the disease. Okay. The next disease is Entamoeba histolytica. So it belongs to phylum Protozoa, subphylum Plasmodroma, class Sarcodina, order Amebina, genus Entamoeba, species Histolytica. So it is also an enteric amoeba of man. So amoebic dysentery or intestinal amoebiasis is formed. So it is um, found in all over the world, like, but mostly in the temperate or tropical regions, we can see more number of cases. So here mostly in Mexico, India, China, and other parts of South America. So it was first discovered by Lambie in 1859. It's a parasitic in nature, and they have lower portion of small intestine, mucus, and submucous layer. So this parasite lives in the small intestine, mucus, and submucous layers of large intestine, and even appendix. Then, in chronic cases, they may enter into liver, lungs, brain, and spleen also, which cause secondary manifestations or symptoms. This parasite is monogenic. That means the whole life cycle it completes within the man itself. So how it looks like? So it is harmful stage is tropozoite. Okay, and the body is covered by semi-permeable plasma lemma, and the cytoplasm has outer clear non-granular ectoplasm, whereas inside the endoplasm there is granular small particles, and the vacuoles will be present, and the nucleus is small and round in shape. Chromatin is present as a delicate ring, and the red blood cells and bacteria are found in the endoplasm. And pseudopodia is present for the movement. And entamoeba has contractile vacuole, which are absent in the isotonic medium. Okay. So this is for the movement of water. Okay. Water goes like in the form of osmosis. So this is the life cycle. So like, sorry. So there are 4 stages of the life cycle of this parasite. That tropozoite, precystic, cystic, and metacystic. So the tropozoite stage is the feeding stage and growing stage, which is the infective stage. Once it grows, then precystic and cystic forms. These are asexual stages. Once it enlarges, it grows, then it forms a cyst. Then finally, the infective stage. So the dangerous stage is propozoid, and this infective stage from one organism to another, so it is mainly transferred through metacystic stage. It forms a cyst to escape the immune system of the other host to like encourage its transmission. So this encystment is nothing but the formation of cyst or transformation of propozoids. So after feeding and growing and multiplying by sexual reproduction, asexual reproduction, this tropozoid comes out of the tissue of the large intestine and forms the encystment. That is cyst. It forms a cyst in the tissue. Now the parasite becomes pericle and food vacuums, everything disappear and it forms minuta form or precystic stage. They secrete few material that is thin cystic wall around them. They have only single nucleus and non-granular cytoplasm, so no granular present, which is called as uninucleate precystic form. Then this nucleus divides into 2 and 4. Now it's called tetranucleate or quadrinucleate. Okay. Now in this stage, a cigar-shaped chromatid or chromatin bodies are present. Okay, these are the features of the cyst. Okay, if you want like which cystic stage means, so these are the characteristics. Okay, and this complete process is called as encystment. It will take few hours. Now the cysts are infective forms of the parasite, and they're resistant to the unfavorable environment. So that's why it forms cyst. They can live up to 5 to 6 weeks even at extremely low temperature and even high temperatures also. They'll be very light, okay, with less specific gravity. So these mature cysts, they pass out into the feces of the man, okay, because from the intestine they come out. They remain there until they get favorable conditions. Now, if this feces contaminates another water or food, okay, through houseflies, cockroaches, or unhygienic handling of the food, now this entamoeba histolytica enters the other human. Okay. In this process, the transmission of cyst to tropozoid again enter. Now it is in the cystic form. Now again they have to go back to the tropozoid form. So when it enters the alimentary canal of the man, so now again this excystment, that is from again cyst, removal of cyst takes place. So the cyst will be dissolved by the digestive juices. And again, this is called as excystment or metacystic stage. Now again, they will form tropozoites. Okay. So a metacystic stage undergoes where a series of mitotic division takes place and 8 nuclei will be formed. And the daughter amoebulae, they pass into the large intestine. They, through mucus lining, again they form mature tropozoites. And this, again, they enter the colon, again precystic form, again same thing, same process takes place. And the life cycle of entamoeba, it completes. So this causes a common disease called amoebic dysentery or amoebiasis, where 60% of the human population suffers. It causes liver, lung, and brain abscess. So that is nothing but the disruption of the cells in this liver, lung, and brain. So here, amoebic dysentery is one disease where blood and mucus will be passed through the stools, and chronic intestinal amoebiasis, where a person suffers from the short bouts of diarrhea, that like some constipation, sometimes diarrhea, sometimes so like that, and dyspepsia to certain foods. They'll be very sensitive to like milk, greasy edibles. Okay, they form gas, flatulence, abdominal discomfort, nausea, all these kind of symptoms. And person carrying these tropozoids, they have normal life. They don't means they may look as normal diarrhea, like sometimes we with the food change or water change, we feel like that. So, but if it persists, then they should consider the doctor. Sometimes they cause abscess. That is, when they enter into the circulatory system, they'll form secondary extra intestinal sites such as liver, lungs, brain, spleen, etc. And they cause abscess of the cells, liver cells, which is also more common, but they have to check the symptoms. Cutaneous amoebiasis, that is on the skin, buttocks or semi-anal region, perianal region, they should check. Preventive measure is proper hygiene. We have to wash vegetables, fruits, use boiled water, maintain cleanliness, wash hands with the soap and water before taking the meal. And community level, so water should be like in the society, like water should be cleaned at particular intervals. They should be checked. All this should be done. Then another disease is Plasmodium, which is malarial parasite. So here it belongs to genus Plasmodium and species Vivax. So we all know this malaria, which is very common human disease. So here malaria means bad air. Okay. So it's common disease from the tropical in the tropical and temperate regions. So mainly it can cause debilitation and death. In now it became less, but usually in previous days, even with malaria, many people used to die. So it is mainly by Plasmodium. So again, it has four species in this: Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, Plasmodium ovale. So Plasmodium ovale is encountered less frequently in Africa and Philippines. Um, they cause different types of malaria, but mostly it is Plasmodium falciparum, most severe. Okay, now it is mainly carried by the mosquitoes. Okay, now efforts are made to develop drugs to control mosquitoes. All these are still going on. Okay, and now for the first time, this malarial parasite in the blood was identified by Charles Leveran and Golgi. He demonstrated this erythrocytic schizogony. So they will burst the cells, red blood cells. Then Sir Ronald Ross, he discovered the oocytes of Plasmodium in the stomach wall of the female Anopheles mosquito. And he was actually, while he was working in the Secunderabad, okay, he was awarded Nobel Prize. So you can see here you have Ronald Ross Institute in Begumpet, okay, where he has actually worked. So August 29th is referred as Mosquito Day. Okay, so here, so Plasmodium, which is digenetic life cycle, that it requires two hosts. First primary host is man and secondary host is female Anopheles mosquito. And this transmission of parasite from person to person through bite of mosquito. Okay, and the life cycle of the parasite usually it alternation of generation. So the asexual reproduction or cycle is by schizogamy, which happens in man. Then sexual reproduction takes place in the mosquito. Okay, so this is alternation. It has three stages: pre-erythrocytic schizogony and exoerythrocytic schizogony and erythrocytic schizogony. So the mature adult form is called as tropozoite and it is amoeboid and uninucleate, and the cytoplasm has granules, small particles, and ribosomes, endoplasmic reticulum, Golgi complex, mitochondria all are present, but contractile vacuoles are absent, whereas food vacuoles are present where they store this food. They contain hemozoin. So in the pre-erythrocytic cycle or schizogamy, this is asexual phase 1. So here the sporozoite parasite, they transfer from invertebrate host to vertebrate host through inoculation. Now, this Anopheles mosquito introduces this to the man with the saliva when it sucks the blood of the human being. After half an hour of infection, the sporozoites leave in the bloodstream. They enter parenchymatous cells of liver, where here 10 days it will take this cycle. In the liver cell, the sporozoid grows, forms round shape, and it is called a schizont. It divides and it forms thousands of merozoites. Okay, this is called multiple fission. Okay, these merozoites are liberated by the rupture of liver cell, and this phase is called as pre-erythrocytic schizophrenia. Here, blood cells are not erupted. Only liver cells will get broken, ruptured, and the cells will come out. Now, in the second phase, again from the liver cells, whatever the cryptozoites they enter, okay, again they enter in the fresh liver cells, again they grow. Here, this is the second stage. Now, the second generation, they are called as metacryptozoites or phanerozoites or metacryptomerozoites. Again, there are two types, micro metacryptozoites or macro metacryptozoites. Micro are small and many in number, macro are big and less in number. Now, again, they enter into the third stage. Okay, they actually become, these merozoites will become more resistant to the immune system, and they remain in the liver for many years until they get the symptoms, clinical symptoms. Now they infect the RBC blood cells, okay, RBCs, okay, for at least 2 years. That means relapse of the disease. Once they get malaria, so it can relapse within 2 years also, because they live in the liver and they can again infect the RBCs. Then third one is third stage, where the asexual multiplication of these erythrocytic schizogamy happens. So here, so whatever these uh, merozoites which are found, they are now enter into the blood, in the red blood cells, they infect, where they grow. This is called a tropozoid phase. So in the tropozoid, a vacuole appears, they increase in the size, the nucleus will be increased. Now they appear a stage called signet ring stage, okay, where the vacuole disappears and they form like a amoeboid structure. Okay, now the cytoplasm of the parasite, they form a black pigment called hemozoin, and numerous granules will be formed. Again, they form erythrocytic. Erythrocytic means they rupture the blood and rosette stage will be formed. Now here the fever start. Once the red blood cells will disrupt, so within 48 to 72 hours, fever start. Okay. Fever reoccurs every 48 to 72 hours. So after this rupture of these blood cells, then numerous parasites are produced and they can sometimes also cause the death of the horse. Okay. Now they have to move to the another host. So after repeated cycles, these merocytes in the RBC, they grow, they form male gametes and female gametes. And now the mature gametes live in the blood, human blood. They remain inactive until a female Anopheles mosquito bites. Now, when a female Anopheles mosquito bites the human being, now they enter into the female Anopheles mosquito. And this entry is usually in the night time. So this is called as nocturnal migration. Okay. Now, in the invertebrate host also, the zygote divides by sexual reproduction, producing enormous number of parasites. And this division of zygote is a peculiar feature of the zygote because whenever it reproduces, it produces a new individual. Okay, that is only once it undergoes asexual reproduction. Okay, then it enters into the mosquito. Within the mosquito, again the gametes are produced, male and female gametes. Microgametes are male gametes, female gametes are the macrogametes. Okay, so now they divide, they form daughter nuclei, and now each daughter nuclei enter into each of this. They'll have the flagella. Okay, they have the cytoplasm. Now they will form a microgametes. Now flagellation also takes place. Now through meiosis, they will form the haploid female gametes and male gametes. Then fertilization takes place and they form a single fusion nuclei which is called synkaryon. Okay, after fusing, means same type of gametes if fused, it is called synkaryon. If dissimilar type of gametes are formed, it is called anisogamy. Once it is done, they form a cyst. Okay, again they remain in the mosquito. Around 50 cysts have been seen in the wall of the mosquito, and again whenever this mosquito bites the human being, again they enter into the human being and they form into the what you call, they form spores and they enter the spores into the bloodstream of the human beings. So in this way the life cycle takes place and the incubation is for about 8 to 40 days. So this is how the life history takes place. So here, like we all know, like what are the symptoms, like intense shivering with cold, high fever, sweating, and we have to prevent the mosquitoes. So this all we know. Like already various measures have taken by the government also. Even drugs are also available, but still research is going on this thing. Okay, I'm just skipping all this because these are the common things. We all know this, and the treatment is also present. Okay, we'll continue the other one, trypanosoma, in the next class. Okay, so you want important questions, right? So, okay, in the next class, I will just let you know. Okay, once I complete this topic. Okay, are there any doubts regarding this? Only one topic is left. Okay, we'll continue in the next class. Okay, thank you so much.
Thank you.